Hello everyone, I hope you're all good. Now in this video, let us study RNA World, the next topic in NCERT text, that is in chapter Molecular Basis of Inheritance. And also we will be focusing on types and functions of RNA. The predominant genetic material is DNA that we have studied from earlier topics. Even though both DNA and RNA can act as a genetic material, that is, they can transfer information from generation to generation. But the question will be, which one is the first genetic material? The answer will be, of course, it is RNA. So that means if you asked with a question, which is the first genetic material? The answer will be RNA. Which is the predominant genetic material in living organism? The answer will be DNA. Then we can ask one more question. What are the evidences to say RNA is the first genetic material? There are many evidences. Here we are going to discuss major two evidences. First one, the essential life processes. The essential life processes include metabolism, translation, and splicing of RNA after transcription process. Metabolism means the total number of chemical reactions taking place in a living cell, that is called metabolism. Translation, synthesis of protein from mRNA. Splicing is removal of introns and joining of exons, and that we will be discussing in detail when we come to the topic transcription in eukaryotes. Here, during the process of transcription, DNA will undergo the process called transcription, that is mRNA will be produced. From mRNA, protein will be synthesized upon the process called translation. Now you could observe both transcription and translation are associated with RNA. And mRNA in eukaryotes contain the coding sequences called exons and non-coding sequences are called introns. Only exons are responsible for protein synthesis. Now, there are enzymes called spliceosomes which will remove the exons and which will remove the introns and join the exons, and that process is called splicing. So now the process will be associated with only RNA. This is the first evidence. Now, RNA used to act as a genetic material, but it also can act as a catalyst. For example, the earlier example we studied, for splicing, spliceosomes are responsible. Spliceosomes are the enzymes which have majorly small nuclear RNAs which are acting as a catalyst. But here, RNA being a catalyst, it is very much unstable. Which molecule will be unstable will undergo chemical changes and structural changes so fast. Hence, RNA will not be a suitable genetic material. Even though it is a first genetic material, it cannot be acting as a suitable for the suitable material for acting as a genetic material. Okay, so here we are clear with the point that the predominant genetic material is DNA, but the first genetic material evolved was RNA. Okay, so now to make the genetic information very stable, DNA has evolved from DNA has evolved from RNA with a chemical modifications, and those chemical modifications are replacement of uracil by thymine and also replacement of ribose sugar present in RNA by deoxyribose sugar. So these two are responsible for stability of DNA. Okay, so now DNA being a double-stranded and having a complementarity, it is suitable for acting as a genetic material. Okay, and also it can resist a changes by the process, by evolving a process called the repair. So repair of DNA we will be discussing in brief in the chapter upcoming topic, that is in replication. So that is DNA repair is a process by which the mismatched nucleotides are removed and a suitable nucleotides will be added to that place. So that process is absent in case of RNA. Now let us briefly discuss the types of RNA. In RNA there are three types. Ribosomal RNA, which is referred to as rRNA. The main building blocks of rRNA are ribosomes, and they play a large role in the process called translation. Translation is a process in which from mRNA there is a synthesis of proteins. Okay, so now the second type of RNA is messenger RNA or mRNA. Messenger RNA is always single stranded and it is involved in protein synthesis, and always messenger RNA is formed from a DNA template during the process called transcription. So earlier we have studied here that transcription is a process in which there is a synthesis of mRNA from DNA. The process is called transcription. So these are the two types of RNA, and the third type of RNA is transfer RNA, or it is called tRNA. tRNA is the smallest type of RNA, that is when we compare the structures of rRNA, mRNA and tRNA, tRNA is found to be a very smallest type. The function of tRNA is to transfer amino acids present in different parts of the cell to the site of protein synthesis. And the site of protein synthesis is ribosomes. Now, it also acts as a link between mRNA and amino acids, hence it is called a adopter molecule. In next video, let us discuss about the process of replication.